In this video, we will discuss what is the relationship between pancreatic cancer and the cholesterol genes. So, in brief, what I am going to cover is that how this cholesterol gene prevent the metastasis of this pancreatic cancer, and this dysregulation of this cholesterol gene will leads to the upregulation of this signaling cytokines, that is TGF beta or transforming growth factor beta. And we have discussed that this signaling of transforming growth factor beta will triggers the metastasis of the pancreatic cancers. And we have covered what are the signaling pathways changes occurred whenever this TGF beta interact with the receptors of the receptors that are present on the pancreatic tumor cells. So in this case, in this video, we will only cover the part that is related to the cholesterol genes. So what happens in this case is that these are the pancreatic cells, and the portion on the upper right side is the blood. So in the regular case, when the cholesterol pathways genes are working normally and there is a cancer cells are presence, what happens in this case is that there is a suppression of this SREB genes. There is a suppression of this NUSREBP1 gene, and this, because of this suppression, this TGF beta 1 will not be able to form. And what happens to the cholesterol pathway genes is that this NUSCRE B2 genes, these are continuously translated into the cholesterol related proteins. And when this conditions is satisfied in the cancerous cells, at that time this classic PDAC will occur. So what does this classic PDAC means? Classic PDAC means this pancreatic cells will be present at a specific site. There is no metastasis of the cancers occurs. However, what happens when these cholesterol genes get dysregulated, and this dysregulation leads to this dysregulation maybe because of this knockout, that is NSDHL. This genetic knockout, or we can say the mutation of this, will leads to the dysregulation of this cholesterol gene. At the same time, it will trigger this NUSREBP1 to upregulate TGF beta 1 cytokines, and simultaneously this statin will also be present in the cancerous cells. In the case that statin is present in the cancerous cells, what happens is that it will inhibit the function of this HMG-CR, and this is necessary to trigger this NSDHL to finally upregulate or translated these cholesterol pathway genes. However, when the statin is present and or there is a genetic knockout of this NSDHL, any of these pathways will get disrupted, so the cholesterol genes will not be able to form. And simultaneously, what happens is that because of the disruption in any of these pathways, this will trigger the upregulation of this TGF beta 1 related genes, and these TGF beta 1 related genes will lead to the release of this TGF beta cytokines, and that will interact with the receptors, and this finally it will trigger this SMAD2 SMAD3 pathway, and that in other terms we can say that it will upregulate the EMT related genes, epithelial to mesenchymal transition genes, and this will cause the basal PDAC or the metastasis of the PDAC. Another way that this NUSREBP1 can be inhibited, that is presented in this paper, is with the help of this LDL. This LDL will interact with this cellular lipid, and this will cause the inhibition of this NuSREB1. So in that case, we can regain in that case we can regain this cholesterol. We can regain this property of the EMT genes from again. We can regain these properties of from being the metastasis of the pancreatic cells to again move back to this classical PDAC, because this TGF beta 1 has been inhibited by this LDL interaction with the cellular lipid. So if you need to understand more about this pancreatic cancer and its relation with the cholesterol gene, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.